Hello friends, today we are going to discuss about the topic of acetylation and methylation. This kind of topic is very necessary to solve many problems, theoretical problems in NET. So before we start to discussion, I have already written another thing that is written here with the help of chromatin remodeling. Why acetylation and methylation is necessary and why we have to read it. So we have to start the discussion. Before we start the discussion from chromatin, we will start it from the nucleosome. Better, it will help to form that nucleosome is the primary structure from where we can easily recognize it, or it will help to clear our concept. Firstly, we know what is nucleosome. If the DNA of eukaryotes is packaged into roughly 11 nanometer pairs called nucleosome, and which consist DNA wound on the surface of histone octamer, then I want to draw a structure that will help. There are octamer structure. Such kind of structure is wrapped with such kind of things. These are the histone proteins, and that is it is DNA. We can write here it is DNA, and these are all histone proteins. So histones are wrapped by DNA, and if we saw here, there are 5 types of histones are there: H1, H2A, H2B, H3 and H4, and these all are present in two form: 2H2A, 2H2B, H3 and H4. Only H1 is wrapped outside like a clip. Suppose if we see a structure, in such a way that will help to form, like if we see a structure, and this is H1, and H1 is present here. H1 is present like a clip outside, but these are all H2A, H2B, H3 and H4 all are present in two form. That's why they are called 2, 2, 2, 2. 8 are present. That's why it is called octamer. So a 11 nanometer of DNA is roughly packaged, nanometers pair of DNA packaged, that is called nucleosome. And these histones proteins are there. There are 5 types of histone protein. I have already told: H1, H2A, H2B, H3 and H4. These all are the basic histone proteins, are highly basic amino acids. Suppose H1, if we suppose the contents H1, H2A, H2B, these 3 amino acids are lysine rich amino acid. These are all lysine rich amino acid, and these 2 are arginine rich amino acid. Arginine, and these 2 amino acid all are basic amino acids. And if we see the content, these are not necessary, but for general information I am giving it to you. H1 contain 29.1% of lysine. H2A contain 11% lysine. H2B contains 16% of lysine, and H3H4 I have already told that it contains arginine containing amino acid. Firstly, H3 contains 10% arginine, and H4 contains 11% arginine. So we have already find here that these are all basic amino acids, and I want to tell you one thing, that DNA is negative and histones are negatively charged. We can say, it is better to say that it is a negatively charged, and histones are positively charged. That's why they become wrapped in a same way. And so we can say, within the nucleosome, charged amino terminal tails bind tightly to DNA. That's why the inside the nucleosome, amino terminal tails bind tightly to DNA. This is the main cause of it. We can easily say this is the main cause of it. And but RNA polymerase, if we start the chromatin remodeling now, but how RNA polymerase gain the access to the promoter, we have already discussed in our lac operon videos. The promoter is an important region where RNA polymerase can bind. If they are closely packed, then how could RNA polymerase bind to them? How could they gain access to attached with the promoter? And nucleosome containing genes that needed to be expressed must be modified promoters. If we discussed that available proteins required for transcription, that is chromatin modeling. If we see that, if it is packed in such a way, we have already seen here and here, they are packed in such a way, so promoter cannot be opened. If promoter cannot be opened, so there is no kind of modification can be occurred. So for this modification, we need a term that is we known as chromatin remodeling, which I have shown here, and it is necessary. And chromatin remodeling can be occurred by sliding the histones or DNA that can express or open up the promoter, which help the RNA polymerase to bind. If some kind of structure, suppose some kind of structure it is here, and expressing part is here, and if it is closed with another part, then how could they express in such form? If such kind of structure is, if the DNA passed through such a way and the region will open, then it can be expressed, and chromatin remodeling can causes such kind of opening things that will help to open up the promoter. That's why the chromatin remodeling can occur. And now we are going to discuss the main topic of today, that is, we have already told that chromatin remodeling mainly happened by two process. One is chromatin remodeling, if we see that happened, it occurs in two ways. One we have already told, that is the main topic of today, methylation, and second is acetylation. Phosphorylation also may occur, phosphorylation also cause, but today we will discuss only these two topics. And methylation can be occurred in two things. One, DNA and another histones. But acetylation can occur only one, that is only histones. That can causes the chromatin remodeling. Now we are going to tell you one thing, that there are almost 3 billion base pair 3 billion base pairs in a typical mammalian genome. 3 billion base pairs are there in a mammalian genome. 3 million base pairs are there in the mammalian genome, and out of 3 billion base pairs, 40% only contains GC base pair, and out of this 40%, only 2 to 7% modified by methylation. These are only methylated. So we can easily tell that 3 billion base pair are human genome, 40% is GC, and only 2 to 7%, this percentage is very low, present in the methylated form. And if we see the structure of methylation, now we have seen a structure. If we see the methylation, that is a 5 prime, and cysteine always bind with the guanine, and in such a way the opposite, it happens in such a way. And here the P denotes the phosphodiester bond. And if I want to give you some information that is very necessary to all of us to know, that why methylation of CpG occurs and what is the cause of methylation. CpG dinucleotide occur less often expected in human genome. We have already seen that only 2 to 7% is methylated part, and as because in course of evolution they mutated into TPG, not they are present in CpG form, they are mutated into CpG is mutated into TPG during the course of evolution. And moreover, the distribution of these dinucleotides is uneven, with numerous short segments of DNA and having much higher density to CpG dinucleotide than other regions of the genome. And these CpG segments are usually, if we see these CpG segment, they are not much more than 1 to 2 kb long. The length of them, if we discuss, that 1 to 2 kb long, and they are very small. In human genome there are 30,000 such islands. If we see the human genome, there are such kind of islands are present, that is almost 30,000. And if we see that 30,000 most of being situated near the transcription site, and this type of structure we seen in the transcription initiation site. We have already told that gene expression is without happening, it will never occur if the expressing region will be closed. If the expression region will be closed in such a way, if until it is not open, it cannot be expressed. So we can easily say that, so it is present in the transcription site, initiation site. We can find that they are present in the transcription initiation site. So methylation is a very important thing, because if we see the methylation produces, if we want to discuss the methylated methylation, generally CpG island, and it is initiation of the transcription site, and And methylation, firstly I want to draw one thing. My discussion is maybe long. CPG 3 prime and 3 prime GPC 5 prime, and methylation occur in the cytosine. Methylation occur in the cytosine, and this kind of structure will not, methylation stop the expression of the gene. Firstly I want to say expression of the gene, stop the expression of the gene, as because such kind of structure may change the architecture of the DNA. The cause I will later tell you, but firstly I want to say where we can found this kind of example. We already know that there are two X chromosome in female, but only the expression occur of one X chromosome. Then what happened to the other X chromosome genes? Because there is methylation occurs in the other X chromosome, that's why they are not being expressed. And now I want to tell you why. The transcription during methylation, when the methyl group added, there forms MECP2 proteins. They form the methylation causes the formation of MECP2 protein, and this MECP2 protein produce in a large amount, and it will compete with H1. Where MECP2 is present, it will compete with H1 histone protein. H1 histone protein I have already told here that H1 histone protein is present here on the outside like a brooch or like a clip outside, and it compete with H1 and it will change the structure, and if it wins the competition, then MECP2 bind to the nucleosome. MECP2 protein binds to the nucleosome, and it will change structure of the nucleosome or architecture of the nucleosome. So gene never been expressed. So gene never been expressed. It compete with H1, and if it binds with the nucleus, if it wins and bind with the nucleus, it will never express the gene, but it will stop the expression of the gene. So methylation, in another word, methylation stops gene expression. We can easily say that methylation stop gene expression, and mainly the protein that will help in it is MECP2 protein. And if there is a mutation occur in, if suppose there is a mutation occur in MECP2, mutation in MECP2, that will form Rett syndrome. Rett syndrome that causes the mental disorder and varieties of muscular disability. So two type of expression I have already discussed. One is methylation, and second we will discuss about the acetylation. Acetylation is the process is just opposite to methylation. If we say methylation, what will be the opposite form? That is, opposite form is acetylation. So we can easily understand, if methylation stop the expression, it will open up the expression, or it will help to express the gene. So now we'll discuss what happened to the acetylation. Acetylation means gene expression. It helps to open up the DNA for replication, transcription. Gene expression mean, I have already told, that replication, transcription and translation. And acetylation is equal to, we can easily say that it is gene expression. You can easily say that it is gene expression, and it will help in this kind of processes. So acetylation causes two things mainly. With the help of histone acetyl transferase, acetylation of lysine 14 in the histone H4. We have already told that there are 5 types of histone: H1, H2A, H2B, H3 and H4, and acetylation occur in the H4. Acetylation occur of the H4 histone protein in the lysine 14. Lysine 14, acetylation occur in the lysine 14, and it is often preceded by the phosphorylation, and this process also causes the phosphorylation of serine 10. This is, I am getting more deeper. So acetylation occur in the H4 gene, and the acetylation, we have already told that DNA is negatively charged, and DNA, we have already told that it is negatively charged, and histones are positively charged. We have already told that histone is positively charged. That's why they are binding to one another. But acetylation causes the histone, it neutralizes the histone. The histone become neutralizes by the acetylation, neutralizes histone. The process is known as acetylation. I have already told that the histone 4 lysine 14 first acetylated, and then phosphorylation of serine 10 occurs, and it causes the neutralization of the histone protein. If neutralization occur, there is no connection between plus and minus, as because one is minus, that is DNA, and histone is positive. So if it is DNA will stay with negatively charged, but it neutralizes, so there is no interaction between them. That's why the DNA remain open, it not been closed. So there is a chance of expression of this site, as we have already told that if DNA part opens up and the promoter opens in such a way, then RNA pol can bind here. RNA pol can bind here. So transcription, translation, replication, all these things can occurs. So we can easily say, now I want to show the picture in such a way that methylation, they have already told here that the transcription off, and acetylation means the transcription, and they are completely opposite process, and I have already discussed why it is happened. And in such a way, we can easily say the chromatin remodeling is the process that is fully dependent on the acetylation and methylation, and we can also say the phosphorylation at the end of the part. We have already seen it here, so we can easily say that the phosphorylation is an important part. So chromatin remodeling, or we can say gene expression is completely depend upon these two process. One is acetylation and another is methylation. And methylation stop the expression of the gene, and acetylation help to express the gene. So thank you very much guys for watching this video. Don't forget to click the like button and don't forget to subscribe my channel. Thank you very much.